Welcome to neurobiology in 5 minutes or less. Right now we're going to be talking about taste part 2. So taste, in the last video we discussed in terms of what sort of mechanisms our body uses to taste different things. We talked about how we have the different papillae, and we talked about the different types of senses that we use in terms of how we taste bitter, sweet, salty, umami, and sour. Those different taste perceptions come from different types of ions, and different ions are stimulating different receptors. So today we're going to start off just with talking about the sweet receptors. So the sweet receptors, just to start off, are a class C receptor. The umami receptors are also a class C receptor, and the sweet receptors look something like this. So just to start out, the receptors in the sweet receptors are a G protein coupled receptor. All that means is that as this receptor is, or as this is stimulated, instead of allowing an ion to flow through or having some sort of direct change through the activation of this receptor, what actually happens is on the inside of the cell, a G protein complex, which will consist of an alpha and then a beta gamma complex, will be stimulated. And those two different things, the alpha G protein and the beta gamma complex, can both go do two separate tasks. And that depends on the different type of receptor and what that specific G protein coupled receptor is stimulated to do. In this particular case, we're looking at we have the transmembrane section of this receptor, and then up here at the top, this part of it, you can see this is the ligand binding complex. So this is where, when we have our ions, they're going to come and they're going to bind here or maybe even here, depending on the different type of ion that they are. And the sweet receptors are not just for sensing glucose. There's a lot of different things that our sweet receptors actually can sense. And these are a few of them. So they can sense sugars, sweet alcohols, artificial sweeteners, which could be sugars that are non-metabolizable, proteins, different types of salts, and different, again, alcohols, but maybe not naturally produced. So all of these different things can bind, and they all would have a different way that they bind to this ligand binding domain. Then we have the transmembrane domain, and then over here, this is where we would have our G protein. And so all of this then begs the question of, how does this get stimulated, and what does that look like? So the way that this would start off is, obviously, we would put something in our mouth. So the first thing that is going to interact with our sweet thing is our tongue. On our tongue, we would then have our papillae. And the papillae would have, depending on which one it is, one or more taste pores. The taste pores are little openings, and then the molecule will go through this taste pore. Once it goes through the taste pore, that is the point at which it will meet the sweet receptor. The sweet receptor will then, if it is a sweet thing that we are eating, it will then bind to one of these sites up here. And then after it binds to one of those sites, we will see some sort of G protein cascade that will ultimately result in the stimulation of a taste cell. It's important to remember that the taste cells here are not, in fact, neurons. They are just regular cells. So the way that they're working is that they will work via this G protein cascade. They will be able to transmit a signal after they have been stimulated. So even though they are not neurons, and even though they do have this G protein cascade, they are not generating an action potential. So it's something important to keep in mind, that there is no action potential generated with taste cells. So that's the rundown of sweet receptors and what they do.